Starfish and sea urchins share a common ancestor with the vertebrates, yet their morphologies appear fundamentally incompatible. To connect the radial symmetry of an invertebrate echinoderm to the bilateral complexity of a fish or mammal, we must identify the biological bridge. Protochordates are classified as a functional grade of organization, encompassing 3 subphyla: Hemichordata, Urochordata, and Cephalochordata. Before analyzing these specific groups, we must establish the strict diagnostic baseline. To be classified within the phylum Chordata, an organism must possess 4 specific anatomical traits at some stage of its life cycle. These include a notochord, a flexible, rod-like structure providing skeletal support, and a dorsal tubular nerve cord situated directly above it. The final 2 requirements are pharyngeal gill-slits, used for filter feeding or gas exchange, and a muscular post-anal tail extending beyond the digestive tract. By cataloging the anatomy of Hemichordata, Urochordata, and Cephalochordata, we can observe these 4 traits existing in varying states of permanence and development. This is most visible in Urochordates, a group that develops these advanced structures in a larval stage only to systematically discard them upon reaching maturity. Decoding the anatomical variations across these 3 specific taxa allows us to reconstruct the foundational blueprint of all higher vertebrates. Hemichordates are soft, unsegmented, worm-like marine animals, represented primarily by the acorn worms. Their body form is divided into 3 distinct regions: an anterior proboscis, a middle collar, and a long posterior trunk. This external division reflects a tricoelomic organization, where 3 internal coelomic compartments map specifically to the 3 body divisions. They possess a hollow outgrowth from the gut called a stomacord, or buccal diverticulum. While previously mistaken for a notochord, current research identifies this as an endodermal structure. Respiration is conducted through numerous pairs of gill-slits located on the pharynx. The digestive system is complete, appearing as a straight or U-shaped tract terminating in an anus. They utilize an open circulatory system, featuring a dorsal heart and 2 main longitudinal vessels. The nervous system is diffuse, consisting of an epidermal plexus of nerve cells that forms 2 longitudinal cords connected by a nerve ring in the collar. Excretion is localized in a proboscis gland or glomerulus. The sexes are generally separate, with fertilization producing a free-swimming larval stage. Hemichordates function as an evolutionary outgroup. They possess gill-slits, but ultimately lack the definitive axial and neural structures found in true chordates. Cephalochordata, known as landslets, serve as the closest living morphological analog to the basal vertebrate archetype. Unlike other protochordates, all 4 diagnostic chordate traits persist without modification throughout the lancelet's entire life cycle. Their body is slender, laterally compressed, and lacks a distinct head or paired appendages. Their musculature consists of thick dorsal lateral muscles, segmented into highly characteristic V-shaped blocks called myomeres. This segmentation works in tandem with a muscular notochord. By preventing longitudinal shortening, it allows the muscles to produce lateral swimming undulations. The feeding apparatus consists of a large pharynx lined with an endostyle and perforated by numerous oblique gill-slits. They utilize a closed circulatory system but lack a true heart. Blood flow is instead maintained by the myogenic contractions of the vessels themselves. The nervous system remains simple, with an undeveloped brain and a complete lack of paired sense organs like eyes or ears. Excretion is handled by a specialized system of paired protonephridia equipped with selenocytes. Cephalochordates demonstrate the stable persistence of the chordate body plan, retaining anatomical simplicity while maintaining the fundamental axial framework through every life stage. Urochordates present a morphological paradox. The adult tunicate, or sea squirt, is a highly degenerate, sessile organism fixed permanently to a substrate. In its adult form, it lacks appendages and a true coelom. The internal cavity is instead a wide ectoderm-lined atrial cavity. Its internal systems are minimal. It features an open circulatory system with a reversing heart, a single dorsal ganglion in place of a brain, and no excretory nephridia. This simplicity is achieved through retrogressive metamorphosis, a specialized strategy where an advanced larva actively degenerates into a simplified adult. Figure 4.31 shows the tadpole larva of Herdmania, featuring a hollow nerve cord and an axial notochord in the tail. Below, the sensory vesicle reveals ocelli and a statocyst, making it an effective dispersal agent. Once the larva attaches to a surface using its adhesive papillae, it begins a systematic anatomical regression. Figure 4.34 illustrates this drastic transition. Phagocytes break down the larval tail, consuming the notochord. The mouth and atrial aperture rotate 180 degrees. Simultaneously, the pharynx enlarges into a filter-feeding basketwork to accommodate the new sessile position. The cerebral vesicle breaks down, leaving only a single neural gland, and the outer test thickens as the transformation into a stationary adult concludes. Urochordates demonstrate that chordate features can be highly transient developmental tools, utilized briefly to ensure optimal site selection before being discarded for a sedentary existence. By synthesizing the traits of these three subphyla, we can map the shared characteristics that link them to the wider evolutionary tree. Their basal affinities link them to the echinoderms. Both groups exhibit radial holoblastic cleavage during embryonic development and form their coelom through an enterocoelic process. They also share patterns of structural asymmetry, reinforcing their shared position within the deuterostome lineage. On the right, we find the definitive chordate markers, the notochord, the dorsal tubular nerve cord, pharyngeal gill-slits, and the post-anal tail. We also observe secondary chordate traits, including a distinct dermis in the body wall and the organized segmentation of the dorsolateral myomeres. Finally, we see homologous structures, such as the endostyle, which provides the evolutionary template for the vertebrate thyroid gland. Each subphylum represents a distinct evolutionary role: Hemichordates as a morphological outgroup, Urochordates as a regressive offshoot, Cephalochordates as the foundational axial blueprint. The Protochordate grade demonstrates the incremental assembly of the vertebrate body plan, providing a series of testable intermediate stages between basal